Well, thanks, Renzo, for the technical support. Sorry about that. Thanks also to Luca for the very kind introduction, for hosting us today. And let's see. I'm very happy to be back here at CMRR. Actually, I did one of these courses, I think, 6 years ago, was it? Oh, gosh. Yeah, maybe. I'm really happy to be back. And so I was asked to give an overview of layer-fMRI. And so this training course is mainly about laminar fMRI tools, and we'll be hearing a lot about that from the developers. And so I'm going to try to lay out some of the concepts that you're going to be hearing throughout this course. I'll give a little bit of foreshadowing of some concepts that you'll be hearing about from the other speakers that hopefully help you to understand why they built the tools the way they did. Okay. So first, I'll start off by defining layer fMRI, which is high spatial resolution fMRI applied to distinguishing functional differences between cortical layers. It's typically applied to cerebral cortical layers, and it's also typically performed at 7 Tesla and above, but we'll see some counterexamples of this. And so why are we interested in fMRI of cortical layers? Well, several cortical layers have distinct functional roles, as Luca had mentioned, and therefore layer fMRI is going to be necessary to map functional architecture within humans. It's also a test bed, I think, for investigating the limits of fMRI. I view it as an extension of the sort of previous program to detect the layout of cortical columns. And I think it's a technology driver. What's good for layer fMRI, I think it's good for fMRI in general, and I think it's going to help us to improve the neuronal specificity of fMRI more generally. But I think what we're really excited about with layer fMRI is that because the different cortical layers are known analytical patterns of feed-forward and feedback pathways, I think layer fMRI can help us to decipher brain circuitry, which really brings this important concept of feedback for the first time into human neuroscience, which I think is really important. And of course, this is going to be key to understanding brain function. And so why a workshop devoted to this topic? Well, it's a relatively new field. It's enabled by increasing availability of these small voxel fMRI studies at 7 Tesla. However, it's pretty challenging, I think, from a data analysis standpoint, which you'll hear a lot about during this course. Now, individual cortical layers cannot be resolved with current fMRI techniques. This is a major challenge, and conventional analysis approaches really aren't appropriate for these data. So we'll hear about these approaches throughout the course. Also, I want to point out there's several kind of interpretational challenges in layer fMRI. We're going to see that there's hemodynamic coupling and spreading across the layers. Also, the functional organization of the layers is largely unknown. Right, so this is really kind of cutting-edge neuroscience that we're doing. Studies are ongoing to kind of understand, what layers are responsible for which tasks. And so currently there's a lack of ground truth when it comes to layer fMRI. But I think this is exciting. It allows us to use these tools to kind of discover new features of functional organization. And so with that introduction, I'll give a quick outline of this lecture. So first, we'll talk about what is a cortical layer, looking at both cyto, angio, and functional architectures. We'll talk a little bit about data acquisition for layer fMRI. I think this is a whole lecture should be devoted to this topic, but I'm just going to point out a few kind of key concepts. I'll talk a little bit about data preprocessing and analysis, again, kind of foreshadowing, I think, what you're going to hear from the other lecturers, and talk a little bit about what's different between layer fMRI and conventional fMRI. We'll talk about the challenges in interpretation and as well as experimental design. And I'll close by presenting a few example applications. As Luca showed, this is a rapidly kind of expanding field. I just want to highlight a few kind of categories of studies that we can perform with layer fMRI. And so we'll start with what is a cortical layer. And so first off, there are layered structures in many brain regions. However, here we're considering only the cerebrum and its cortex, okay? And so cortex is Latin for bark. And so as the name implies, its laminar organization really is kind of a defining feature. The most of this bark is neocortex, which is evolutionarily the most recent. It comprises the esocortex, which has 6 main layers with a rough common layout. The rest is allocortex, which has fewer than 6 layers. We have paleocortex and olfactory cortex, archicortex, such as hippocampus, which is the oldest in either 3 or 4 layers. And then other types of cortex in the transitions between these types. And so even though this sort of basic 6-layered esocortex has a layout that's common across the cerebrum, and we'll see that there's important regional differences as well. Here you can see that, for example, motor cortex is missing its layer 4. So it's not really potentially a full kind of 6-layered cortex. And so these regional differences across areas are going to provide us with a basis for defining cortical areas based on their laminar anatomy. Okay, so you can see here, this is a beautiful work from Ramon y Cajal, that there's a very clear banding or layering of the neurons to appreciate, in these examples. Here I'm showing, again, cells near to the white matter at the bottom, cells near to the peel surface at the top. You can see both in this sort of radial direction from the white to the peel surface, as well as this tangential direction, there's a grid-like structure, both in terms of the morphometry and kind of organization of these neurons. So it's quite clear that there's some layered structure. And so there are various anatomical definitions of layer types in the isocortex. They can be defined based on cytoarchitectonic features such as cell morphometry and density, as well as myeloarchitectonic features such as myelin content and fiber patterns. And so this isocortex exhibits these 6 common layer types shown here, as well as many sublayers, also based on cyto or myeloarchitectonic features. And here you can see some examples of these various subdivisions. Now, while these 6 basic layers or layer types are roughly present across the neocortex, I've heard it said that the 6-layered neocortex or isocortex is really a unicorn. The number of layers that can be distinguished really varies a lot between areas and differs between different histological stains. And so it's quite uncommon to see a given cortical area that has exactly these 6 layers, like in an example of motor cortex that I showed you previously. Okay, so just to point out a few of these layer types, again, here we can see the 6 layer types across the isocortex, a few ones that should be pointed out. With layer 2 is the external granular layer 4 is the internal granular layers, because it has these really small kind of neurons look like grains of sand. And this layer 4 is quite special. We call it the granular layer because it's sort of the most prominent. Any layers that are above this granular layer we call supragranular. Layers that are below this granular layer we call infragranular. So you start to see that, with layer-fMRI as well as from the cytoarchitectonics, there's sort of like 3 kind of super layer types that we can kind of consider. Supragranular, granular, and infragranular. These correspond to superficial, middle, and deep cortical layers we often refer to in layer-fMRI. Generally, we don't have small enough voxels to be able to resolve these layers, but this kind of 3 super layer kind of scheme actually does capture a lot of the important features, both anatomically and functionally, which I'll talk about in a few minutes. Okay, a few other important layers here. We have the external band of Baillargeau, as well as the internal band of Baillargeau. This external band of Baillargeau is also known as the stria genari in V1, which I'll talk about in a moment. Also, we have this plexus of Exner, which has a lot of apical dendrites, some inhibitory cells, axonal terminations, some cortical feedback is located here. This band of Exner is kind of funny. My PhD supervisor used to say that this layer 1 is basically a copy of the white matter found at the top of the cortex. A lot of like horizontal fibers there, very few neurons. Okay, we'll see that it's very rare that we actually see any kind of neuronal activity up here in layer 1. I'll come back to this in a few minutes. Okay, so again, this layer 4, this band of Baillargeau is also known as the stria genari. When it's in V1, it provides us with an anatomical kind of definition of striate cortex. Here you can see the line or stripe of genari, this heavily myelinated band of tissue in layer 4B of V1. We can see this ex vivo. We can see it in vivo often. We can also see it in fixed human tissue. What's quite remarkable is that you can see it here with the naked eye. This is a slice from Jonathan Horton's lab. If you kind of zoom in a little bit here, you can actually see the stria quite clearly. I think it's probably the most kind of apparent kind of microanatomical feature of the brain, which is why it was discovered, in the 1800s. Quite easy to see even with a simple microscope. And so the cortical area borders are defined based on their laminar features. We have the famous cytoarchitectonic atlas from Brodmann, other myeloarchitectonic atlases, for example, from Elliott Smith. Now, the area borders in many of these regions are quite distinct, but other regions are pretty subtle. And so these parcellations can be somewhat subjective. We see that the cytoarchitectonic and myeloarchitectonic divisions roughly agree, but not perfectly. And so it's still kind of unclear what's the best way to define the cortical area. We can talk about this more at the coffee break if you're interested. Okay, again, so there's these roughly 5 cytoarchitectonic-based types of isocortex. We have Agranular cortex here on the left, such as M1 primary motor cortex, which doesn't have this granular layer, all the way to a granular cortices on the right. We see that primary sensory areas, such as primary somatosensory, primary auditory, primary visual, are all examples of this sort of granular cortical area. You can see that there's very different cortical types distributed across the brain. And we think that there may be distinct functional roles of these different cortex types. So this is some work from Lisa Feldman-Barrett, who is sort of proposing that maybe agranular cortex, dysgranular cortex, granular cortex may play different functional roles in these multi-area kind of brain circuits. And so we think that form follows function. There may be kind of a reason why we're seeing these differences in microanatomical structure across these areas. So note that there's varying thickness of the cortical layers across areas. Again, here's M1, which either has an absent layer 4, depending on who you ask, maybe it's a really thin layer 4. You can see here that the layers really shift around across the cortical areas, and the thickest layers are around 500 microns. What this tells us is that, again, in order to be able to resolve individual layers, we're going to need like 200-ish micron voxels, and that's really something we can't yet achieve with fMRI. Also, it shows us that these layer positions really shift around a lot across cortical areas, even between adjacent areas, V1 and V2. Layer 4 is sort of jumping quite abruptly. So in laminar fMRI, again, we very rarely, if ever, fully resolve the layers, and I think this is important. Now, if I sort of normalize this by cortical thickness, we can see again how thick layer 4 is in V1 compared to the adjacent cortical area V2. Again, what this tells us is that if we're using a kind of a fixed resolution for our laminar fMRI, we may get a bias towards seeing kind of more activity from layer 4 in V1 compared to the adjacent cortical area V2. And so again, this is a source of sampling bias because layer 4 again its thickness varies quite a lot across cortical areas. Okay, so this is a course on layer fMRI or laminar fMRI. You may hear other terms in the literature, such as depth-dependent fMRI, layer-specific fMRI. You may also hear depth-resolved, cortical depth analysis. So what are we talking about? What's the cause for this distinction? Well, it's because there's sort of a non-trivial relationship between cortical depth and cortical layer. We know that the layer positions and thicknesses, as I showed you, really varies a lot across the folding pattern. This is shown by Van Economo. He showed that in sulci, the upper layers are expanded, and in gyri, the lower layers are expanded. This is also pointed out quite famously by Bach, who also showed that this induces kind of an exaggeration of the geometry of layer 4. We see that layer 4 is sort of pulled down in sulci, kind of pulled up in gyri, kind of, again, a very non-trivial relationship between cortical depth and the position here of layer 4. And we'll come back to this. This is addressed with the equivolume principle, which you're going to hear a lot about throughout this course. I also want to point out that, again, as I showed you in those bar graphs, we see discontinuity in layer positions, even between adjacent cortical areas. You can see this in both cell density and myelin stains. And so these jumps are most frequently sort of identified in kind of primary sensory areas. I think they're less identified in association cortex, but still you can see here that there's a kind of a complicated mapping, again, between depth and layer that also varies between neighboring cortical areas. So the question is, how do you know the position of layer 4, for example? It's not always at 40% depth. It can be at 50% depth, depending on the cortical area. And so what do? You got to look it up. You got to look up, generally you have to look up the depths of the layers for any given cortical area. It's not consistent, even between V1 and V2. Now, these layer features are detectable with MRI, especially with these anatomical techniques with higher resolution. Sometimes you can see contrast between the infragranular and supragranular layers. These infragranular layers tend to have a little bit more myelin. They appear a little bit darker, especially in T2 star weighted acquisitions. We can see these ex vivo. We can see some of these features increasingly in vivo. But generally speaking, we have to kind of infer the locations or the depths of these different cortical layers, from our cortical segmentation. You're going to hear a lot about that throughout this course. Okay, so let me talk a little bit about the functional roles of the cortical layers and sublayers. And here you can see some examples of lumping and splitting of these cortical layers. So for example, layer 4 can actually be subdivided into sublayers and sub-sublayers, for example, and we know some of these sublayers have really distinct roles. For example, layer 4C alpha is on the magnocellular pathway responsible for motion and depth processing, part of the WARE pathway. Layer 4C beta is part of the parvocellular pathway, which is responsible for processing form and color, part of the kind of the WHAT pathway. You can see here in the visual cortex, layers 2 and layers 3 kind of have very similar functional roles. So you'll hear, vision people talking about layer 2, 3, because it's understood that these two layers kind of have very similar functional properties. So again, lumping and splitting. Here's another example. You can see activation across the different cortical layers. These are just two examples. Any kind of feature that you guys notice, that's sort of shared between these two plots? Now, what about the layer 1? Layer 1 isn't even shown in these plots because it doesn't really have much, functional role, especially in vision. As I mentioned before, layer 1 has very few cells, okay? And so this is really important. Sometimes when you see a paper and they talk about, activity in layer 1, you sort of question, whether maybe this is due to some sort of bias from the vasculature that we'll talk about later. But generally speaking, we're not expecting to see much activation in layer 1. Now, perhaps the best known property of cortical layers is that specific layers are the sites of inputs and outputs along so-called feed-forward, feedback, and horizontal pathways. And many people look to this seminal paper from Fellman and Van Essen for this general kind of scheme or ontology. So they identified across looking at many different studies. This is 1991, but it's already kind of a meta-analysis from very kind of classic anatomical studies. They identified several different pathways. They identified an ascending or feed-forward pathway that was characterized by an origin being either supergranular or bilaminar and a termination being into layer 4. They identified a lateral or horizontal pathway in which the origin was bilaminar and the termination was more columnar. And they also identified a descending or feedback pathway in which the origin was infragranular or bilaminar and the termination was multilaminar. So again, just by looking at these patterns of connections, we can get some idea about which areas are communicating to each others and which pathway is being engaged. So this led to, this kind of stereotype pattern led to some theories about, some potential kind of canonical connections between connected cortical areas, such as the canonical microcircuit from Douglas and Martin, as shown here. So here's an example, again, the kind of updated version of the canonical microcircuit. You can see here that, forward connections generally project into layer 4. We see these backward connections, these feedback connections projecting to layers 2, 3, and layers 5, 6. I've left out some details. There's a little bit more going on in this canonical microcircuit. We see kind of recurrent excitation and inhibition, within these layers. But you can start to see what I mentioned before, this sort of 3 kind of super layer scheme we tend to use in laminar fMRI does capture, some of these, functional properties. We can see that the superficial depths, tend to be sampling from layers that are responsible for these feedback connections. The middle depths tend to capture layers that are looking at these forward connections, and the deep depths tend to capture layers that are, again, looking at these feedback connections. Okay, so how do we relate laminar structural and functional connectivity? And so, in this last course we did a couple of years ago, I spoke right after Josine, and I had the slide up, and she had made a few quotes that I wanted to capture, so I quickly threw them on the slide. Josine reminded us that neurons span multiple layers, and neurons are assigned to a layer based on the location of the cell body. Okay, and so you can see here, again, these pyramidal cells in layer 5, they have these apical dendrites that can span many cortical layers. And so what does this mean? So if you can imagine, if we have an input anatomical projection into the superficial layers, then when this pathway is engaged, in which cortical layer do we expect to see the neuronal activity, right? We may expect to see the neuronal activity, in the soma. We may expect to see most of the energetics happening, again, here in layer 5. And so what this means is that there's some complexity in terms of relating these anatomical projections to where we'd expect, the functional activity to occur. Now, what's remarkable is that, there's also very distinct angioarchitecture of across the cortex that changes a lot across area borders. Even has been proposed that there's different vascular layers. So this is a seminal work from Henri Duvernois, who identified 4 vascular layers across the esocortex, including this sort of vascular layer 3, dense, band of capillaries that kind of coincides roughly with cytoarchitectonic layer 4. And what's remarkable about these angioarchitectonics is that they also vary quite a lot across cortical areas, especially in the visual cortex. You can see here, just to orient you, here's the calcarein sulcus in humans, the white matter. You can see that this dense band of capillaries sort of ends abruptly at the V1, V2 border. You can almost identify or define V1 based on its angioarchitectonics. We see this in primary visual cortex. We also see this at the M1, S1 border. And these sort of conspicuous differences in angioarchitecture across the cortex has actually led some people to propose an angioarchitectonic atlas, being able to define different cortical areas based on their distinct angioarchitecture. Now, this is quite difficult. It's quite difficult to look at the blood vessels for reasons we can talk about offline. This is work from Pfeiffer. This was in macaque cortex. So this is a kind of a cool photograph of these angioarchitectonic fields painted onto a wooden model in the lateral and medial views. Again, pointing out that we see that there are kind of really conspicuous differences in the vascular architecture, so much so that we can define cortical areas based on this architecture. And just to point out that this kind of distinct or conspicuous angioarchitecture extends beyond the cerebral cortex. Just one quick example, looking at a layered structure in the thalamus, the lateral geniculate nucleus, we can see in this vessel density map that we see high vascular density that corresponds to the different cell layers of the LGN. And so it kind of makes sense. We know that the vasculature is there to deliver energy to the neurons, and so it makes sense the vascular architecture would kind of resemble kind of the energetic needs of the cortex. And we see that the vasculature is really adapted to the function of the brain. This is work from Bruno Weber in Nikos Logothetis' lab, looking at the correlation between kind of cell density across the cortex and vessel density. Saw a pretty decent correlation between the two. But he saw an even stronger correlation between cytochrome oxidase activity. So cytochrome oxidase is a mitochondrial enzyme. We see it sort of more pronounced in regions that have higher baseline metabolism. We saw a very strong correlation between cytochrome oxidase and vessel density. And so what this tells us is that it's quite cool. It tells us that the brain's functional architecture is sort of reflected in its vascular architecture. I think this is really nice. It tells us that we can kind of learn a lot by studying the vascular architecture to see kind of a little bit about patterns of neural energy demands in the cortex. I think this is sort of bad news for fMRI or any hemodynamics-based technique, because it tells us that we may see stronger hemodynamic responses, not necessarily because there's stronger neural activity, but maybe because of the sort of, for example, increased capillary density. And so that's something you're going to hear a lot about throughout this course. And so also we have really intricate intercortical vascular anatomy. I showed you in one of the earlier slides a sort of grid-like structure of the layout of the neurons. We also see kind of a grid-like architecture of blood supply and drainage in the cortex. So again, intracortical vascular anatomy, just to orient you, we have these large PL veins that kind of drain deoxygenated blood from large cortical domains. You're going to be hearing a lot about this. This is where most of the deoxyhemoglobin is, and that's where we see the largest sort of bold effects. And so you'll hear a lot about how we can account for this in our laminar fMRI data. We have these ascending venules that run perpendicularly across the cortex, also kind of troublesome for laminar fMRI. And again, here in the parenchyma, this sort of randomly oriented capillary bed, where presumably the spatial coupling between the hemodynamics and the neural activity is the tightest. And so the idea, again, is that fMRI voxels at different cortical depths are going to sample from different levels of this vascular hierarchy. So just as a reminder of the pathways of blood supply and drainage, we have some symmetry breaking in the cortex. We know that this oxygenated arterial blood is supplied downwards from the surface into the tissue. Again, I'm plotting the surface at the top, the white matter boundary at the bottom. Most of the oxygen passes from the blood into the tissue at the capillary bed, and this deoxygenated venous blood is drained upwards from the tissue up to the surface. Now, one big question in high-resolution fMRI, certainly layer fMRI, is where are the control structures for blood flow regulation? It's sort of a central question. For many years, it was thought that it was mostly these smooth muscle cells that kind of aligned to these diving arterioles, which would enable kind of blood flow regulation at the scale of individual cortical columns. It looks like we see smooth muscle cells all throughout these diving arterioles, which suggests that there may be blood flow regulation at the laminar level, as well as control structures on these precapillary arterioles, which again would be able to provide laminar level control. There's still some question about whether there are control structures along capillaries, but I think for the purposes of laminar fMRI, not quite so important. We do see that there are control structures in place that could deliver blood to specific cortical layers. Now, just as a reminder of the timing of the hemodynamic response and how we see coupling of hemodynamics across depths. So first, immediately following neuronal activity, we see blood flow increases in the capillary bed. This could be due to active dilation of capillaries, but I think it's more likely to be due to pre-capillary arterioles. Immediately afterwards, we see upstream propagation of dilation up to the intracortical arterioles and up to the pial surface, again, to deliver fresh blood to the tissue. And then a little bit later, we see downstream changes in flow and oxygenation in intracortical venules and pial veins. And so what this means is that even with fine scale regulation, changes in blood flow or volume or oxygenation are going to be coupled across the cortical layers. And this is something that we need to take into account. Okay, so next I'm going to talk a little bit about data acquisition for laminar fMRI. Again, just a few words because this is a pretty big topic. There's a lot of challenges in high resolution fMRI acquisition, mostly because we're really at the limits. We're kind of pushing the scanners to their limits in terms of what we can achieve in terms of sensitivity, as well as spatial and temporal resolution and coverage. Now we see that there's different capabilities across sites, which makes this kind of a hard challenge to address. Generally, we see different capabilities across different vendors, different instrumentation setups, software, sequence implementations. Non-standard hardware and software are often needed to acquire these high-resolution data. Specialized workflows, protocols are required. I think another thing, especially for newcomers to layer fMRI or high-resolution fMRI, I think a lot of newcomers don't really know what to expect, in terms of image quality. Like, what does a good image look like? What does a bad image look like? And so I think this is something that we can, as a field, I think help out with to show people what their expectations should be. If their image looks, too ghosty, we should be able to provide them with an example of what their data should look like in ideal circumstances. Now, it's quite difficult because in order to achieve high resolution, we have to balance between several different kind of competing trade-offs. Generally, what we're looking for is spatial resolution or small voxels. However, in order to achieve high sensitivity or image quality, this typically competes with temporal resolution. If you want really high spatial resolution, you have to encode more lines. The acquisition becomes a little bit slower. And also, it competes with spatial coverage. You can achieve very high temporal resolution with a small number of slices, but if you want more coverage, you're going to have to sacrifice temporal resolution, and in some cases, spatial resolution as well. And so these three kind of factors are competing for resources. It's not always possible to simply kind of crank up the resolution to achieve a desired voxel size. We need to balance, again, these trade-offs. There's also many nonlinearities in scanner performance, several sweet spots in terms of what the scanner can achieve. I remember on the classic 7T, we used to be able to achieve a 1.1 millimeter isotropic acquisition. And it was kind of conspicuous. Why did you guys pick 1.1? Well, we didn't. It was actually much easier to achieve 1.1 compared to 1.0. Again, sometimes you see sweet spots in terms of the scanner performance. And so we need to identify those in order to make best use of the scanner. There's other considerations. Image reconstruction times are getting longer and longer. Scanner instability, vulnerability to physiological noise and motion, contrast, shine through, safety limits. It's pretty tricky to achieve the desired resolution and coverage. And so what do we do? Like, how do we address this? Well, my recommendation is it's really tough to optimize a protocol from scratch. Okay, I think that the best advice I can give you is to start with an existing protocol that ideally has been optimized already and has been used successfully in the past. I think starting from scratch, can take a lot of time and effort, to balance all of these trade-offs and all of these nonlinearities in terms of the scanner performance. So this is a recommendation for those newcomers. Now, considerations in choosing an fMRI acquisition, I think there's two main considerations. One is the imaging readout, and the second is the form of functional contrast. Now, there's many different imaging readouts to choose from these days. The sort of classic is 2D echoplanar imaging, or EPI, with SMS or multiband. Now we see greater adoption of these kind of advanced 3D kind of echoplanar imaging techniques. It's called echoplanar because it's typically one shot per kind of KX, KY plane. Sometimes you see multiple shots per KX, KY plane. And so this is a very kind of efficient acquisition that I think every high resolution lab should have access to, a good 3D EPI sequence. Classically, we've seen, and also more recently, we've seen some people use, 2D flash, which can achieve, really high resolutions, although with kind of, again, longer acquisition times, so lower temporal resolution. We're also seeing some new 1D line scan acquisitions. This is sort of the ultimate trade-off, really high resolution in one direction, say like 200 microns along a single line, but just one line of the brain. So very low spatial coverage. And we can see that it also can achieve really high temporal resolution, which we see in several kind of specific kind of advanced applications. Okay, so that's imaging readout. Functional contrast is also a situation in which we need to consider various trade-offs between sensitivity and specificity, as well as reliability and robustness. And so bold or gradient echo bold is sort of the default, has really high sensitivity, but really poor specificity. However, it's universally available, really easy to use, quite robust. Other techniques such as ISL to measure cerebral blood flow has really low sensitivity, but really high specificity. It's really kind of weighted towards the microvasculature, the capillary bed, which is where we want to be measuring. Yet it's pretty, somewhat available. It's really challenging to use, especially at high resolutions and at 7T, and so something that's not commonly used in layer fMRI. Vaso, which is a measure of cerebral blood volume, has sort of a nice kind of balance. It has kind of moderate sensitivity, pretty good specificity, and I think it's becoming more widely available, yet I think still requires some assistance to use. It's hard enough to optimize, your imaging protocol for spatial and temporal resolution and coverage. Vaso, I think, also requires some optimization, especially for kind of non-standard brain regions. And if you have any questions, Lorenzo is, of course, the expert, and he can help you get your Vaso protocol set up for your brain area of interest. Okay, so as I mentioned, the biggest issue with this gradient echo bold is its lack of specificity, and you're going to hear a lot about this large vein bias in gradient echo bold versus non-bold fMRI. And so here's an example of this large vein bias. These are data from Renzo. So here in blue, first we have the CBV response to neural activity kind of plotted across cortical layers. We can see this sort of classic kind of double peak activation, which you'll see many times throughout this course, which we can kind of observe with Vaso, but it's not quite seen in the gradient echo bold. It's kind of smeared out across depths. We see this kind of large activation near the peel surface, which we call peel bias. This is likely reflecting the fact that at the peel surface, we have these big kind of draining veins that kind of collect blood from all of the cortical layers. And so what this means is that, while gradient echo bold is nice, it's easy to use, it has really high sensitivity, but really kind of low specificity. Vaso, however, has lower sensitivity, so you need to average a lot more data, but higher specificity. And there's other contrasts, which I mentioned, have different trade-offs. It's interesting. So the original layer fMRI studies actually used bold. I think most of these studies, actually, David had mentioned to us that a lot of these studies used 3D grace. However, Vaso is increasingly used for layer fMRI. A few more comments about these downstream hemodynamic effects in bold. So even if there is arteriolar control at the laminar level, for bold fMRI, we still have to condemn these sort of downstream effects. And this was sort of nicely illustrated by this kind of paper from David Norris's group. So the idea here is that, as I mentioned, the deoxygenated blood is sort of drained from the tissue up towards the surface. And so what that means is that if we have activity in layer 6, we expect to see some bold response, around layer 6. And that's going to be drained upwards. We're also going to see some bold in layer 4. And this gets even worse if we have more activity, more neuronal activity in the top of layer 4. We expect to see kind of an integration or an accumulation of the bold signal in the superficial cortical layers. So this kind of integration or accumulation of the bold response across layers is indicated by this bar plot. We can see that if we look at, for example, layer 2-3, some proportion of those bold signal changes are actually going to reflect, bold that, originated in layer 6. This can be modeled by so-called, laminar spread functions that try to convey how the bold response in any one cortical layer is going to be seen in the upper cortical layers as it's sort of drained up towards the surface. And so again, these bold or venous responses in upper layers are going to reflect integrated or kind of an accumulated response from all of the lower layers, which also contributes to this large peel bias. Okay, so next I'd like to say a few words about data preprocessing and analysis. Again, this is sort of the kind of focus of the course, but I just wanted to kind of prime you for a few concepts that you'll be hearing about. So first, I'll talk a little bit about data preprocessing. Now, this is often viewed as kind of a dry topic, pre-processing, but just to sort of motivate it, here's a study that was quite interesting, published in Nature, no less. So the organizers took an fMRI data set and distributed it to 70 independent groups and asked them to all analyze the data, and they collected the results and kind of compared them. And as you might imagine, there's a lot of variability in the reported results across these 70 groups. And they said that the strongest factor that contributed to this variability was spatial smoothness in the data processing. And this spatial smoothness arose from analytical steps beyond explicit smoothing. So this is due to, again, different groups had different pre-processing techniques, which contributed inadvertent smoothing, which kind of contributed to this variability of the results. And so inadvertent smoothing, especially in the context of high-resolution fMRI, is really problematic because it counteracts. We spend a lot of time balancing these trade-offs to get to our small voxels. It's no pity to kind of throw away that spatial resolution, by using kind of suboptimal data preprocessing choices. And so why is preprocessing different for high-resolution fMRI or for layer fMRI? Well, I think for a couple of reasons. First of all, conventional preprocessing techniques fail. If you use a conventional preprocessing technique, you may see that it just does not work with your data. And so there's some practical challenges. But as I mentioned, we cannot afford to lose any of our kind of hard-fought spatial resolution. Our voxels are really at the edge of being able to distinguish the responses across depths, even between these three sublayers. And so the question becomes, how do we preserve the high spatiotemporal resolution of our imaging data? We've written a couple of papers on this, and you'll be hearing a lot more about this, throughout the course. But I just wanted to point out, a few examples of ways in which you could, potentially achieve higher resolution through your preprocessing. So first, conventional motion correction on high-resolution fMRI data can often fail. So here's an example from my lab. We have a kind of a high-resolution API. This is a subject who happened to not really move very much. Good for us. And on the right is the same data, this movie, after conventional motion correction. And this isn't a cooked example. A postdoc came to me and said, "Jon, what's going on with these data? What's happening?" And we realized that it was actually due to the kind of standard motion correction that they applied to the data. Now, I think of motion correction as really kind of comprising two steps. The first step is estimating the motion. And you can see here in this movie, and it loops a little bit, you can see that it looks like there's some false positive motion detection. The subject really wasn't moving, but you can see that the correction is actually kind of bouncing around a little bit. This is perhaps due to the high levels of noise in these EPI data, as well as there's, the second step is a correction, which can also fail. You can see here the default parameters that they used resulted in really noticeable blurring of the data. You can see that one frame, there it is a much sharper. Again, the default parameters, not only introduced motion, but introduced blurring as well. Now, this particular software package has settings that you can use to kind of mitigate at least some of these blurring effects. But again, standard techniques aren't going to work well on these high-resolution data. My recommendation is to use AFNI. I think it does a good job in terms of both estimation and correction. But again, the point here is, you can't necessarily use the same workflows that you use for conventional fMRI data, especially in these cases where we're really trying to preserve spatial accuracy and resolution. Now, I should also point out that in conventional preprocessing pipelines, we'll apply many transformations to the data individually, including kind of rigid transformations such as motion correction, as well as non-rigid transforms such as different forms of distortion correction. And then you can see here, so I'm plotting here the additional blurring induced by these transformations into the fMRI data. So you can see a lot of blurring that's actually being induced from these transforms across the brain. This is because every time we transform the data, we need to resample the data back onto the image grid. And this resampling requires interpolation, which is going to smooth out the data. And if we do 3 consecutive steps of resampling, we kind of interpolate the data 3 times, which introduces a lot of blur across these fMRI data. Now, we can mitigate this somewhat by using a higher order interpolation. It doesn't get rid of the problem, but it does reduce the blurring quite a lot. Now, I think that sort of a best practice approach that was advocated by the Human Connectome Project in a paper that I was involved with, over a decade ago, is to compose these transformations kind of mathematically. So again, we compose them so that we can perform a single resampling, which reduces the amount of blurring. Again, the idea is that it's kind of hard to not interpolate your data at all, right? I think you generally have to do at least 1 resampling of your fMRI data. The point is to try to limit that to the single resampling to avoid or at least minimize this blurring. Okay, I want to talk a little bit about laminar fMRI data analysis. Again, you're going to be hearing a lot more about this from the other speakers, but just to kind of foreshadow a little bit. So what's different? What is different between, layer fMRI analysis and conventional fMRI analysis? Well, the big thing is the main step in layer fMRI analysis is to compare activation across depths, right? In order to infer laminar differences in activity and laminar differences in function. And so I think the key piece is that we need to know which layer is each of our fMRI voxel sampling. And so on the one hand, layer fMRI is easy, just to look at superficial depths, look at deep depths, compare the activation. But it's really hard to know exactly which depth your fMRI voxel is sampling. We're going to hear a lot about these problems throughout the course. Again, we're often kind of inferring or estimating layer positions from our anatomical data. And so, of course, there's going to be differences between the anatomical data and the functional data in terms of contrast and resolution. So it requires really careful geometric alignment of the fMRI data to the anatomical data where these layers are being defined. But again, the layer positions are inferred from these anatomical data. They're not measured directly. This is very different from columnar fMRI, right? In columnar fMRI, you need to, there's no anatomical, prior that you can use to define, the column borders. But laminar fMRI is kind of easy in some sense, upper layers on the top of the cortex, the lower layers on the bottom. But it's still quite difficult to really understand, exactly which, layer your fMRI voxel is sampling from. Now, in laminar fMRI, we typically don't perform much in the way of spatial smoothing. We also don't perform any spatial normalization. We typically instead will average these laminar activation profiles across our subjects, which is a form of fixed effects analysis. And so we'll be hearing more about that throughout the course. But the big issue is, I think one of the biggest challenges with layer fMRI is the accuracy of cortical segmentation and surface reconstruction. Again, this is what we use to kind of define the cortical layers, and it still doesn't work as well as we would like, especially for these high resolution, anatomical data, and especially with the accuracy that we need, to identify these cortical layers. So again, there's several schools of thought that I think you're going to hear about, throughout this course. The first sort of school of thought is to perform an automatic surface reconstruction from your anatomical reference, such as NP-RAGE, and then distortion correct VPI. This is quite challenging. There are some newer EPI techniques that have less distortion, such as kind of highly segmented or accelerated 3D EPI, but there still can be some distortion. One can also apply the distortion to the anatomical data. But the point is that the automatic segmentation and cortical surface reconstruction can often fail and can often have some inaccuracies. And so that's something that many people try to address. Another school of thought is to perform an automatic cortical surface reconstruction from the EPI data themselves. Actually, last time I was here, I think Cheryl was a big fan of this approach. So we have an EPI acquisition that has the exact same distortion, exact same readout as our fMRI data, but has kind of anatomical weighting that's more conducive to kind of segmenting the cortex. So you can have an EPI with T1 weighting, an EPI with magnetization transfer weighting. This is really nice because at least gets rid of the problem of aligning the fMRI data to the anatomical reference because they both have the exact same distortion. The problem is that they both have the exact same distortion, right? And so the idea is that these anatomical references are going to have geometric inaccuracies. We're going to see distortions in the cortical thickness. And this is going to result in, again, kind of misestimation of the cortical layer locations, especially in regions with strong distortion. And so because of these problems, because we don't really have anatomical automatic segmentation techniques that really work robustly and provide us with the necessary accuracy, many people will manually segment, like literally kind of draw the borders of the cortical gray matter in order to reconstruct surfaces either from the EPI data themselves or from the NP-RAGE. And so, oftentimes when people really desire this kind of high level of accuracy, this is an attractive option. I think when people first start to approach this technique, they ask me, they're kind of a bit anxious about, well, am I going to get it right? I'm not sure how to define these, borders, worried about subjectivity, I'm worried about bias, inaccuracies. And also it can be quite time consuming. It's one thing if you have a kind of a small, kind of localized region that you're trying to segment, if you're trying to extend this across the entire brain or do this across multiple subjects, again, it can be quite impractical. But you see that this has been advocated because the automatic techniques don't work as well as we would like. And so this is, I think, a big issue in layer-fMRI that we as a community, I think, need to address. I think Anna might be mentioning there's a few of these new kind of machine learning tools that do, I think, a much better job, but maybe not quite evaluated on our 17 data yet. And so the point is, cortical segmentation, which is a prerequisite for defining the layers, I think isn't working as well as we would like today. Now, generally, we start with this cortical segmentation, and then we can define cortical depths to each of our fMRI voxels. Again, we're going to calculate a cortical depth at each fMRI voxel based on the distances to the surfaces that define the cortical boundaries. And so here's how this kind of voxel-based cortical depth assignment works. We start with a segmentation of the gray matter, a surface at the peel boundary, a surface at the white matter boundary. If we consider our fMRI voxel grid, for any given voxel, we can calculate its centroid and then calculate the distance from the peel surface and the white matter surface to assign a depth to each of these voxels. So again, we're using surfaces, but we're assigning depth, using this kind of voxel-based approach. And so generally what we do then, once we have these depths assigned, we're going to generate depth profiles by kind of pooling across multiple voxels. And just to show you how this works, so this example, this is just looking at intensity profiles across an MP range. So you can imagine here, so if we have, say, 6 voxels that are spanning the cortex, and if we look at the image intensity at this one location, shown here in green, you can see that there's roughly kind of 6 kind of unique intensities across the cortex. Okay, makes sense. And now if we look at the same cortical depth profile, but at a different location here at the red line, you can see that there's a very different kind of pattern. The transitions between these different unique intensities are occurring at different cortical depths. And so you can kind of see where this is going. So if we sum across multiple of these locations, in this case 25 locations, the transitions between the intensities are going to occur at different cortical depths. And so if we average them all together, you can see kind of a nice kind of smooth image intensity across depths. Now note that this approach assumes that all voxels within an ROI have similar intensity or activation across the layers, which is allowing us to average in this way. Now, in order to do this, we need enough locations. In this case, we have 25. If we only looked at 3 or 4 locations, you'd imagine this profile would look very irregular. So in order to avoid sampling bias, you need to, again, look at many different locations, pool over a large enough ROI. So in this example, if we look at a really small ROI and consider the cortical depths sampled from the voxels within this ROI, you can see that there's some biases. We have more voxels that are sampling from the peel surface compared to other cortical depths, and that's a form of bias. We can reduce this bias by increasing the size of the ROI. As we go to larger and larger ROIs, we're going to more evenly sample the cortical depths. But you can see there's a trade-off here. Again, another bias variance trade-off. If we make the ROI size too big, this assumption that at every location within the ROI, we're going to see similar intensities or activation patterns may be violated. So the big question then becomes, how many layer bins should we extract during the analysis? And so this is something that's, I think, quite counterintuitive. So even with 0.8 millimeter voxels and a 2 millimeter cortical thickness, you can define many, many cortical layers depending on the size of your ROI, depending on how much folding there is within the ROI. You can see here that if we go to 2, 3, 4, 5, even 10 layers, by bidding into more layers, we can reduce the aliasing effects of pooling within this ROI. And a big question I'd like to come back to later is, this comes up a lot with reviewers. A lot of reviewers are like, gosh, you have 0.8 millimeter voxels and a 2 millimeter cortical thickness. Like, how is it that you're able to divide up the cortex into 20 bins? And so it's because, again, each location is kind of sampling a kind of different subset of cortical depths, and by pooling across this large ROI, we can resolve this sort of kind of nice, kind of smooth laminar profile, which again, I view as sort of eliminating aliasing that would be seen if you only looked at one cortical location at a number of depths. Oh, note that, okay, so this plot, we're looking at cortical depth. As I mentioned before, there's kind of a complicated relationship between cortical depth and cortical layers. And now I want to kind of give you some insights into how we account for this in the analysis. So you can see here that, as I mentioned before, we see that the sulci, the upper layers are expanded and the gyri, lower layers are expanded. And so this kind of led to this kind of very kind of attractive idea that there may be some principle that kind of dictates the positions of these layers such that volumes of tissue are kind of preserved across the cortex. And so this led to a so-called equivolume principle that people tried to use in order to predict layer positions from the geometry of the cortex. So here's an example of this equivolume principle used to predict layer position, also known as kind of layerification. So here shown on the left, so if we solve the Laplace equation to try to estimate the geometry of layer 4, the Laplace equation kind of minimizes curvature, kind of smooths out, these curved structures. And so if we use the Laplace equation to try to predict the position of layer 4, it really does a bad job. It really is not, capturing the fact that the geometry of layer 4, again, is exaggerated as we look across the cortex. If we use the sort of kind of simple equidistant approach in which we basically say, okay, layer 4 is at 40% of the cortical thickness at every cortical location, it doesn't do such a bad job. It's okay, but it doesn't vary with the folding pattern as we know the position of layer 4 does. It's this anatomically inspired equivolume principle that performs best, is able to capture the position of layer 4, because again, it sort of adjusts the depth at which it assumes layer 4 is sitting at, based on the local cortical curvature. And it's this approach that's used in many laminar fMRI studies today. So here's an example. This was provided to me by Renzo many years ago, I guess, looking at the depths predicted from the equidistant approach and the equivolume approach. You can see that there's subtle differences. Of course, the two are very similar in regions where the cortex is nice and flat, but in regions where the cortex is curved, you can see that there's some discrepancies or differences between these two predictions. And so if we look at cortical depth profiles between the equidistant approach and the equivolume approach, you can see that, at least in this case, we really see small differences between the two approaches for typical sub-millimeter voxel sizes. You can see that the cortical depth profiles, again, from the equivolume and the equidistant are pretty similar. Now, I think that we sort of know that the equivolume approach is capturing a feature that's known to be there. It may be that this approach is going to work best for really small voxel sizes. I think for conventional fMRI voxel sizes today, I think that one doesn't have to fret too much about which approach you use. I think both are going to work pretty well. I also want to point out that with layer fMRI, we're typically pushing our voxel sizes to be as small as they can possibly be, and so sometimes people want to be able to perform some smoothing of these data in order to kind of win back SNR. And so when we do this, we sort of we don't want to perform kind of conventional, kind of three-dimensional volumetric smoothing. We want the smoothing to kind of be anatomically informed and sort of conform to our priors about the activation pattern. And so this sort of so-called anatomically informed smoothing, which I think you're going to hear about later today, is one such way to kind of smooth in such a way to kind of preserve activation within cortical layers. And so if we have a prior, if we assume that the activation should be consistent within individual cortical layers, we can perform this two-dimensional or laminar smoothing. If we have other different hypotheses about the layout of the functional activation, we can perform other forms of smoothing. Again, these are kind of steerable kernels that sort of track the folds of the cortex. And again, this is a really nice way to kind of think of it in terms of coloring within the lines, making sure that the smoothing is kind of contained within individual cortical layers in order to avoid smearing out activation between layers. And so here's an example of smoothing within cortical depths, again, from Renzo. I think what's important here is that when we're smoothing within depths, we're imposing a prior. We have this prior knowledge that if the activation really is consistent within layers, we're sort of allowed to smooth within layers. But that's, again, another form of a bias variance trade-off. So here you can see with these unsmoothed data, it's a little bit hard to see, kind of visualize the patterns of activation. If we perform this sort of layer-specific smoothing, we can better visualize the layer-specific activation patterns. You can see this across two different sessions. Again, I think this is really important to show the activation maps before and after the smoothing. I think of it in terms of, if you've got like a scatter plot of some noisy data, like two variables, and you want to kind of visualize, the relationship, you can fit a line to kind of understand like how these two pieces of data are related to one another, and that can help you summarize or visualize the trends. But you wouldn't just show the line. You want to show the raw data that was used to kind of fit the line in order to get some sense for the variance in the data to make sure that the model that you're fitting, is capturing the trends in the data. And so I strongly recommend, showing the data before and after smoothing, just to make sure that the smoothing isn't inducing any patterns into the activation. Now, a big question in layer fMRI analysis is how to address this draining vein problem. You're going to hear a lot about this. There's several different kind of categories of approaches. One is to perform vein masking from a venogram. Again, just identify veins using some anatomical data and just try to like throw out fMRI voxels that intersect these veins. This approach does not account for many important effects. We know that the draining vein effect varies in bold fMRI with the distance from the vein, the orientation of the vein relative to the B0 direction, the diameter of the vein. And so this approach is, quite simple to perform, but I think does not account for many important effects. Another way to address the draining vein problem is to just ignore, throw out voxels at the PL surface. This is another prior. We know that the biggest veins are on the PL surface. We can just toss those out. Of course, that means that we're losing a lot of data, and it doesn't account for those pesky ascending veins that I mentioned to you before. There's also several deveining models that you're going to hear about, later in this course. I like this kind of categorization from Renzo: additive models, multiplicative models, convolution models. There are many approaches to try to address this draining vein problem. I think generally speaking, none of these work perfectly, and so this is again a big issue with gradient nickel bold, and one of the main motivations for pushing beyond these bold acquisitions for layer fMRI. Okay, I'll talk a little bit about challenges in interpretation and experimental design. There are many challenges in interpreting these laminar fMRI data. As I mentioned, we know that there is vascular biases, as I mentioned, high capillary density in layer 4, for example, as well as hemodynamic coupling across the depths, which makes interpreting these fMRI data challenging. It's also challenging to know what layer or layers is a given fMRI voxel sampling. I think in order to interpret these data, it requires prior knowledge about what the roles of the layers are, which are the input layers, and which are the output layers across different pathways. Because when we try to infer whether, for example, we're seeing activation that's consistent with, say, feedback, we need to know which layers are receiving that feedback. And the big question that we talked about a bit at dinner last night, again, is how to validate these measurements in humans. I think we often don't get enough credit for using kind of non-invasive techniques. With invasive measures, there's many different ways to kind of validate these observations, but there are fewer options available in humans. I want to stress the importance of experimental design. So again, from Renzo, this is a classic study. I think one of my favorite aspects of the study was that actually used multiple different tasks in order to elicit distinct profiles from the same cortical region. So here Renzo used tapping with touch. You can see this sort of famous kind of double peak response. He also used tapping without touch, shown in green, touch only, shown in black, kind of ipsilateral tapping, shown in red. And for each of these tasks, he could observe sort of a very different profile of activation across the layers. I think this is really important because this helps to argue against the activation profile, this double peak profile, reflecting biases at any particular depths. And so I think that one way that we can kind of mitigate, or at least kind of control for some of these confounding factors, is through kind of clever experimental design to really make sure that we're not seeing effects that are simply reflecting vascular architecture. Also, again, as I mentioned, not a lot is known about neuronal processing at the laminar level. So it makes it difficult to design novel stimuli that are appropriate for fMRI that we know can induce top-down or bottom-up processing. There's a lot of complexities in stimulus design. And as I mentioned, it's often not clear which cortical layers are going to respond to our stimulus, our task. As mentioned, these anatomical connections are often insufficient to predict the activation patterns. And intercortical networks are really nonlinear. Effects can really propagate rapidly from one layer to the next. And so the peak neuronal activity can occur outside of the input layer. And so just to showcase, two laminar fMRI studies that looked at top-down effects to kind of indicate some of the complexities in interpreting these data. So in this one study here at the top from Lars Muckli performed a decoding task and showed that there was peak performance shown in these superficial cortical layers. And another task using these inducers of illusory contours performed by Peter Koch observed a strongest activation in layer 6. And both of these studies attributed the observed effects to feedback pathways. You can see that in one study, maybe the feedback pathway was engaged in layer 1. Another study, the feedback pathway was engaged in layer 6. It's hard to know maybe ahead of time, like which layer is going to be activated with a particular stimulus or task. A lot of complexities. and these designs. Okay, so I'm probably, all right, let me just wrap up with a couple of example applications just to kind of give you a sense for how we can use these layer fMRI techniques. So a few popular kind of categories of applications of laminar fMRI. I think the most popular is to try to detect whether feed-forward or feedback pathways are engaged by a stimulus or task. This can be used with using, or this can be tapped into using attentional modulation or try to engage top-down processing. Also, people are trying to study different aspects of functional architecture, such as stimulus preference or tuning, which really vary a lot across the cortical layers, as well as laminar functional connectivity, for example, from resting state fMRI data. I think this is quite exciting because it allows us to potentially infer the directionality of information flow from different cortical areas. So again, the idea here is that if we look at the correlations between cortical area A and cortical layer B as a function of depth, we see a strongest correlation between output layers of area A and the input layers of area B. This sort of suggests that information is flowing from area A to area B. And so using this kind of depth information allows us to potentially infer the directionality of these correlations. Now, many layer fMRI studies are focused on a primary sensory or motor cortex. We're seeing a trend toward looking beyond these primary sensory motor cortices. We see recent laminar fMRI studies in prefrontal cortex, ventral visual cortex, into language processing. I think this is a sign of the maturity of the field, that we're extending these techniques beyond kind of simple sensory paradigms into more kind of cognitive neuroscience. I think this is quite exciting. Also seeing some recent clinical research applications of laminar fMRI, for example, an ALS patient on the left and dystonia on the right. I think it's going to take some time before these techniques are really robust enough for kind of routine clinical research. But again, I think this is a sign that these techniques can be providing useful clinical information. As I mentioned, I've been focusing on kind of the cerebral cortex and cortical layers in the cerebrum. However, laminar fMRI can be performed in layered structures outside of the cortex, such as in the retina, hippocampus, olfactory bulb, LGN, superior colliculus. Again, very similar tools and techniques can be extended beyond the cerebral cortex. And so just to wrap up, what do reviewers of laminar fMRI papers want? What do we see? What are the patterns we see? Now, I think that for those of you who are going to be using kind of non-bold fMRI, the question is, will the reviewers want a comparison to gradient echo bold? I think no. I think we're past this. I think at this stage, these techniques are kind of been used enough that you don't have to be interested to hear what Renzo thinks. I mean, I think this is no longer required, but we can discuss this over coffee. I think reviewers want to see individual steps. There's recognition that laminar fMRI is hard. And they want to see, for example, they want to get convinced that you're defining the cortical layers properly, that you've registered your fMRI data to the anatomical data well. There's this big question about the number of layers relative to voxel size and thickness, as I mentioned. I think a lot of reviewers will kind of balk at using kind of 10 cortical depths with a 0.8 millimeter voxel and 2 millimeter cortical thickness. Is this something that you should be aware of? I think also reviewers want some justification for voxel size. I think as a community, it's not clear what voxel size do we really need. Is maybe 1.2 millimeter voxels might seem too big. Can this be used to look at activity between superficial layers and deep layers? Maybe. What's the resolution that we need? I think there's not really consensus about this. I think reviewers want to see results in individual subjects. That's because studies today still tend to use small numbers of subjects. They want to see consistency across the individuals. This may change as we're moving to kind of higher end studies. Again, we're seeing a trend towards increasing N. And maybe we can ask the course faculty if anything else that they see. This is something that I see quite often, and perhaps we can discuss this at the coffee break. Okay, so just to kind of sum up differences between laminar fMRI and conventional fMRI, I think basically everything is different. Acquisition, pre-processing, analysis, interpretation, experimental design. Our hypotheses, again, with laminar fMRI, really doing some cutting-edge neuroscience. We're kind of getting beyond what's sort of known about human brain function. And so I think that this is both exciting. Intuition can fail. Sometimes if you have a stimulus design you think is going to engage a top-down pathway, it's may not be as kind of easy as it may seem. And so I think there's some challenges here, as well as validation. I think that Yeah, this is a kind of a thorny issue, how to validate, especially when animal models really aren't going to provide us with the exact same patterns of activation. And so this is something that we as a community, I think, need to think about more. Okay, so let me wrap up a few takeaway points. So I showed you that EsoCortex has 6-layer types, right? But not all areas have 6 layers. And this kind of 3-layer or super layer scheme that we use a lot, and laminar fMRI captures many relevant aspects of function. We infer the layer positions from segmented cortical gray matter, but the segmentation can fail and layer positions shift across the folding pattern. So this is quite tricky. I've shown you how layers are the inputs and outputs of feed-forward and feedback pathways. I think we tend to see most of the kind of input signals in fMRI. We tend to see kind of less activation in these output layers. But again, it's quite exciting because this provides us with some ability to kind of bring feedback, the concept of feedback, into human neuroscience. But again, not all connections and pathways are known. The canonical microcircuit doesn't really apply to every cortical area. It's kind of a useful kind of summary, but still a lot to be sorted out. There's many challenges in data pre-processing. Again, we need to retain our high imaging resolution. A lot of conventional techniques will fail. Data analysis is still non-standard for layer fMRI, but we're going to hear a lot about this in the next presentations. And just to end, a lot of challenges in interpretation and a big need for clever experimental design. Again, to isolate the effects of interest, establish controls, as I showed you, or to establish a null hypothesis. As I mentioned, vascular architecture is quite cool. It's really similar to functional architecture, but this provides some compounds in terms of any kind of hemodynamic-based technique. Also, I showed you that there's coupling of hemodynamics across cortical depths, which we need to contend with. Vascular biases, incomplete knowledge of neuronal function, and validation is missing. So lots of challenges, but a lot of excitement too. I have to say that I'm pretty impressed by how rapidly this field has evolved. Quite exciting what we can do with these tools. I think you're going to learn today about how to do these laminar fMRI analyses the way we do them today. I wanted to give you these ideas to help you think about how we can make these tools even better, how we can push laminar eye into the future to maybe address some of these challenges, and again, extract more information about the human brain with this powerful technique. Okay, so I just want to thank funding support, team in the lab. Big thanks to Luca, Renzo, and Anna, of course. Thanks, you guys, for your attention. And if there's time, Luca, happy to take some questions, and then we can also discuss these topics at the coffee break. Thanks.
Do you guys have any pressing questions?
Not pressing, but yeah. I think the few things that you showed how the functional organization across layers correlates with the vascular and myeloarchitectonic layers. So what do you think about using data-driven segmentation? So we could get possibly vascular data from like QSM or something, or look at myelin mapping techniques to look at data-driven segmentation or estimation of layer profiles. So is there work?
Yeah, no, I think, well, what I would say is it's relatively straightforward to segment kind of large vessels and maybe mid-sized vessels. It's difficult to segment the smaller vessels. For example, it's harder to estimate kind of capillary density in order to see these features of angioarchitectonics, partly because, again, these vessels are much smaller than the voxel size. In humans, capillaries like 8 microns in diameter. I'm not going to resolve those anytime soon. So these QSM techniques are great, because they're sensitive to iron and myelin. It's hard to tease out these kind of vascular effects from QSM. And so I think that there are some techniques that people are looking into to try to infer capillary density. I think that's still pretty hard, generally again, because it's hard to distinguish the blood signal from the tissue signal. And of course, we want to be careful of any circularity in terms of kind of segmenting, identifying from the fMRI data themselves. And so that's challenging. But I love the idea of trying to, again, right now we're guessing where the layers are. I would love to see where the layers are. Exactly. And so, how do you do that? As I mentioned, there's some hints at being able to distinguish between infragranular and supragranular layers. Layer 4 can be seen in a couple of the different cortical areas. I think that has promise. It's difficult to, I think it's pretty easy to see the striatum denari in V1. It's hard to see it everywhere. You can just, here's a voxel layer, I see it, but to really have it be detected reliably across all of the visual cortex, that's hard.
Yes.
And so, yeah, I would love to see more tools, that can identify the cortical layers anatomically, data-driven approaches, but I think we need to be thoughtful of the analysis, as well as, the best analysis technique in the world might not work if you give it kind of crappy data. We need to kind of work together to try to find better acquisition techniques that can be used with these more kind of powerful segmentation tools. Yeah, thanks for your question.
Thanks, that was a really fantastic overview. You mentioned there's not neurons in layer 1, but one thing that we do know is in layer 1 is these really large apical tufts.
Yeah.
So in the presence of things like calcium spikes there,
Yeah.
a lot of the feedback hypotheses that people usually propagate rely on feedback to that particular layer. So I'm wondering how layer-fMRI can address that.
Yeah, it's a great question. Yeah, so Josine mentioned the same thing, I think. Yeah, we do see that it is a site of a lot of cortical projections, right? And so layer 1 has a role. The question is, from the perspective of fMRI, where we're really looking at, these blood signals, where would we expect, the changes, the metabolic changes to occur that would be correlated with changes in blood oxygenation or blood volume? And so that's when I think it gets a little bit harder to understand. Do we expect to see hemodynamic changes in layer 1 when there's input from these cortical projections? Unclear. I mean, I think that I would love to, first of all, layer 1 is really thin. Layer 1 is right up against the CSF. It's right up against these PL veins and PL arteries. So it's actually one of the most challenging, layers to measure with layer fMRI. And so I'd love to understand this better. I think some of these kind of line scan techniques, you might be able to kind of resolve this. But I think right now, yeah, maybe this is an example where we can kind of talk to our friends in kind of invasive, electrophysiology and hemodynamics and get some more ground truth. What would we expect to see in layer 1 in terms of hemodynamics? But yeah, I think it's a great question, but yeah, we might not be there yet. Yeah, but yeah, let's talk more about that. That's a great point.
No other thing. Can I go?
Oh, yeah.
Yeah.
Well, I mean, if people want to take a break, I mean, I guess,
Yeah,
I won't be offended if you want to grab a
I think the
coffee,
questions
but
are
I'm welcome
relevant
to.
for everyone else. I mean, coffee is available outside. If you guys are craving it, you can go. But we recommend listening to the question because it's really when you learn the most, in my opinion. But it's up to you guys.
Yeah.
I mean, it's free.
Yeah, we can keep it informal. Yeah.
So you also pointed out that basically the neurons are they extend across the whole layers, but you see to define the layers based on the cell body, so to speak, right?
Well, yes and no, right? I mean, these cytoarchitectonic definitions are based on the cell body.
Yeah.
These myeloarchitectonic definitions are based on myelin. And you see like correlations between the two, but they're, generally the myelination occurs like sometimes a little bit above, the cell bodies. And so there's mappings, people have proposed mappings between the two. But again, mostly, yeah, cytoarchitectonic cell bodies.
So my question was around basically the way we try to interpret fMRI based on the neurovascular coupling. There was this paper by Logothetis, I think in 2001, where they tried to
Yes.
argue that this is correlated with LFPs, which is more corresponds to the postsynaptic activation as compared to the basically firing of the neurons.
Yes.
So would that help interpreting the microcircuit model that we have? Because we know that the signal we're getting is basically post-synaptics.
So that's a seminal study that I think everybody in fMRI needs to be aware of. It was like the first study to perform electrophysiology alongside of fMRI. And we learned a lot about that. I mean, but that was 25 years ago. And it's, there's some subtleties. But yes, I think it kind of makes sense that, because, spiking is kind of energetically kind of downhill process. A lot of energy goes into sustaining membrane potentials and, repolarizing cells like after they spike. It does make sense intuitively that energetics would be more kind of tightly coupled with kind of synaptic activity compared to spiking. So again, some subtleties, but I think the idea makes sense. But the question becomes, again, there's so many kind of connections, like so many pathways within the cortex. Sure, you can imagine if there's a thalamocortical projection that projects from the LGN into V1, you might expect those recipient cells in the granular layer are going to, exhibit, like large changes in their synaptic potentials, large changes in LFP and therefore kind of large changes in fMRI signals. But then those cells are going to immediately project, to layer 2, 3. That also going to project down to layer 5, 6. And so on the one hand, you may say, well, this energy that we're seeing consumed in V1 is reflecting kind of neuronal activity in the LGN. But at the same time, there's again a lot of processing going on within V1 as well. And so, yeah, there's some subtleties to this interpretation. I think for many years, people thought that fMRI kind of only represented input because it was kind of reflecting synaptic activity. But I think that it's a little bit more subtle. There's many kind of connections, many synapses within V1. And so this kind of input activity is going to very quickly be transformed into kind of local activity, which is also going to cause large, changes in energetic, demand and supply and hemodynamics.
Thank you so much.
Yeah, no, thanks. No, that's a cool point. I can talk all day. Yes, please.
I'm from the field of social neuroscience.
Great, great.
So my question is, there are relatively few studies targeting the subcortical
Yes,
areas or regions or
yes,
networks.
yes.
So what are the things that we should consider when we are doing using layer-fMRI on the subcortical area?
Okay, I can think of a couple of things we need to be concerned with. I think that these subcortical regions are, tougher to image, first of all. They're kind of further from our detectors, less SLR. They tend to have more kind of physiological noise. So I think just fMRI in these subcortical structures, can be different. We see that the hemodynamics are different. The vascular supply is quite different. For example, we were talking yesterday about, some studies in hippocampus in which we're seeing that, the vascular supply in hippocampus is really quite remarkable. And we're still kind of learning how to deal with, these peel vein effects in hippocampus. We're still trying to figure it out in the cerebral cortex. It's even harder. A lot of these subcortical regions, like the inferior colliculus, is like really highly vascularized. Other regions, the neurovascular coupling seems to be like quite different. Like an olfactory bulb is like very different neurovascular coupling compared to the cortex. So I think there's many issues in terms of these confounds, in terms of neurovascular coupling, vascular artifacts. I think that these structures aren't all layered structures, of course, as well. And so I think less is known potentially about, the kind of functional architecture of these regions in humans. I think a lot of the tools that we have really aren't built, for these subcortical structures. Like how do you perform like a layer analysis in hippocampus? There are these new tools to unfold the hippocampus, which are very new and I think, quite exciting. I think, fewer tools available for kind of brainstem regions. And so, yeah, I think, lemon or fly is hard enough. The conventional tools don't apply. I think for these subcortical regions, there may not even be tools that are in place today. And so, let's see what else. Yeah, hard to get the data, hard to analyze the data, hard to interpret the data. Yeah, but I think we just, but there are lots of studies. I think that there's more and more fMRI studies in the subcortical regions, of course. If we really want to understand, brain circuitry, we can't, leave out, thalamus, brainstem. So yeah, more work in this direction, but yeah, more work is needed.
Sounds hard.
Yeah.
I'm gonna add to that, just because it's hard doesn't mean you don't have to try. Everything is hard, but when things are hard, in my mind is a sign that we're moving in the right direction because everyone can do easy things, not everyone can do the hard things. So maybe the good questions are always difficult. layer-fMRI is, like Jon has been pointing out, is difficult to begin with. But there are a couple of papers, the one by Renzo we're working on as well, and from David Pember's group, we're starting to look at, for example, hippocampus and other subcortical structure at the layer level. So hard for sure, unless you have a 10-5 like us, it becomes easy. But I think they're very important questions, so we need to get
Yeah. Yeah, thanks, Luca. I mean, on the one hand, yeah, I think it's important to point out like what directions, what areas need work. But yeah, I don't mean to say that one shouldn't dive in. I mean, there's been a lot of like really beautiful data in these subcortical structures. But I think that, again, if anybody wants to engage, they need to be aware of the fact that a little bit of an uphill battle. But still, I think a lot we can learn from studying these areas.
Right. Thank you so much. Let's thank the speaker again. All right, let's take a maybe 20 minutes break and be back at 22.